Okay, welcome back, everybody. So today we're going to continue talking about our regulation of mean arterial pressure. So when we left off, we had been talking about the fact that our mean arterial pressure is a regulated variable, regulated through homeostasis. We don't want it to drop too low. We don't want hypotension because then we don't have enough blood flow to organs. In particular, if you don't have enough blood flow to, say, your brain or your heart, it's an immediate threat to you and your life. We also don't like our mean arterial pressure to be too high or have hypertension because that's also a problem over the long term as it adds stress to our organs. So we know that our mean arterial pressure is responsive to our heart rate and stroke volume, because those are our cardiac output, our flow. And we know that it's also responsive to our total peripheral resistance. So we left off right before starting this reflex mechanism called the baroreceptor reflex. So we're going to walk through that right now. But just so you know, right, our whole point of this reflex is to make sure that when our blood pressure, our mean arterial pressure, experiences a sudden drop, we want to get that mean arterial pressure back up, right? We want to maintain blood flow to all our important organ systems. So especially we want to make sure that we're feeding the brain, feeding the heart. So this is going to start out by your body sensing a drop in the mean arterial pressure. So we have arterial baroreceptors in the arch of the aorta and in your carotid sinus. So sensing that arterial flow coming right out of the heart, and also checking again that pressure going up to your brain, up through the neck. Okay. So these can sense a drop in mean arterial pressure. So the way they do that, right, is that they have essentially a sense of how stretched out the walls of those arteries are. Right, so they're always sending action potentials up to the brain, up to our cardiovascular integrating sensors in the medulla oblongata, right? And if our blood pressure drops, if our mean arterial pressure drops, that means those blood vessel walls aren't so stretched out anymore. So those action potentials that they're sending up to our central nervous system kind of slow down, right? Because we're responsive to the stretch. So your brain is going to know that this means that we've seen a sudden drop in the mean arterial pressure. Okay. So our cardiovascular control center is then going to respond. So overall, our response is really going to be regulated by our autonomic nervous system. Pulling us back up is really going to be the job of the sympathetic branch of our autonomic nervous system. But we also have to make sure that we turn down any parasympathetic activity. And remember, whenever we're thinking sympathetic, parasympathetic, we've always got this kind of trade-off. It's like a dimmer switch rather than a light switch, just one or the other. So we're turning down our parasympathetic activity. And we know that that parasympathetic activity, really the one place that's going is to the SA node in the heart, affecting the heart rate, right? So as we turn that parasympathetic activity down, our SA node sees a rise in heart rate because we're not suppressing those action potentials in that pacemaker anymore. The sympathetic activity is working alongside that, right? So parasympathetics are going down, sympathetic activity is going up, okay? So we likewise see an effect on that SA node in the heart, working hand in hand with that parasympathetic activity dropping to bring up the heart rate. We know that the sympathetic nerves go more places. So those sympathetic nerves are also going to the ventricles of the heart, making them squeeze harder, increasing their contractility, increasing stroke volume. So those are two factors that make up our cardiac output. So we've just raised our cardiac output here, okay? The sympathetics are doing more stuff. Okay. So specifically, we're paying attention to what's going on in the veins and the arterioles as part of this response. So that's the kind of new part here for this unit, right? So we know that in the veins, they have this state of the walls called tone, right? So they have like a little bit of tension in the walls of a vein, not much at baseline, but when we see these drops in mean arterial pressure, we're going to lift up that venomotor tone a little bit. We're going to put a little more tension in the walls of the veins, which means they start to behave a bit more like our arteries. So that's a drop in compliance, right? Compliance is a relationship between volume and pressure. So even with the same volume of blood in them, we're now going to see that venous pressure rises a bit. So venous pressure does create a gradient between the veins going into your right atrium and the heart, right? So we can create a high-low pressure by rising up that venous pressure coming back to the heart. So our result here is we have more venous return to the heart, more blood coming back to the heart, raises our end diastolic volume. Right, so the heart fills with more blood because we increased this pressure gradient between the veins and the atrium. Okay. So we now see an effect here, which is to raise stroke volume, because when the heart is a little bit bigger, a little bit more stretched out, that brings it actually to its optimal size if we're thinking about like how stretched out sarcomeres are ready to contract enough. So we raise our stroke volume with this increase in venous return. Our final piece, in the arterioles, big thing that our sympathetic nervous system is doing is increasing vasoconstriction. Right, so we know that our arterioles have alpha adrenergic receptors throughout them in skeletal muscle and in the heart. We also have beta 2, but our big point of concentration is those alpha receptors here. So vasoconstriction in response to norepinephrine means that we have an increase in our total peripheral resistance, right? Because a constricted arteriole with that smaller radius has higher resistance. We add it all up, we've got a higher total peripheral resistance. Yep. So their overall effect to raise our mean arterial pressure then, can't have both these on at once, okay. To raise our mean arterial pressure is because we multiply these things together to get that mean arterial pressure. All right, so we raised all of them. Our mean arterial pressure goes up. This will be a bit of a negative feedback loop. Once we're back up to a reasonable mean arterial pressure, we don't have to keep doing this. Good. So we turn off this response once we've returned to normal. Yep. Questions about this process? Okay, if you think of any, let me know. Okay, so this is in particular a response to hypotension, to that low blood pressure, because that's a more immediate threat to life. But we also want to realize that this is really just kind of a compensation for low blood pressure, for these drops in blood pressure. We haven't necessarily changed what's going wrong overall. For example, think about you would see this response not just when you stand up and have a little low blood pressure going to your brain. You'd also see this response if you're hemorrhaging, right? Now, obviously, you can't fix a hemorrhage just by raising your cardiac output and increasing your resistance a bit. You still have to solve the problem. Okay. So we've got this quick compensation for our change in blood pressure, but it doesn't necessarily correct our problem. If we want to long-term kind of change things, in the case of a hemorrhage, you need to actually stop the bleeding. But even after that, long-term regulation is going to have a lot to do with the kidneys. Going to have to do with managing the blood volume, right? So managing physically how much fluid there actually is in your bloodstream. Okay. That links up a bit to our hormonal controls. Okay. So when we're thinking about regulating mean arterial pressure, using our hormones. One of these is hopefully pretty familiar, our epinephrine. You can already kind of predict how that's going to play a role in affecting our mean arterial pressure. And I've got two more hormones that we've mentioned briefly as kind of an add-on stuff that affects the cardiovascular system, are vasopressin and angiotensin 2. So when we think about effects overall on MAP, we're mainly thinking, with a few exceptions, about how do we bring MAP, our mean arterial pressure, up to where we want it to be, right? Because of this extreme immediate danger of low blood pressure. So epinephrine is going to increase our mean arterial pressure. And we kind of know how it does that, right? So we know that it acts on the smooth muscle of our arterioles. Overall, our effect of epinephrine here is to increase our total peripheral resistance. We do know, though, that effect is a little dosage dependent, right? So if we had a low versus a high amount of epinephrine, we'd see different effects in the body. We'd see the blood getting redistributed a lot as we are raising that total peripheral resistance. We're doing a lot of vasoconstriction kind of throughout the body, but we know we're also routing more blood to the heart and skeletal muscle. But overall, we're increasing our total peripheral resistance. We're also increasing our venomotor tone, right? So those veins are stiffening up a little bit. So we know that through that flow of effects that we just saw in the baroreceptor reflex, we're increasing our venous return, ultimately increasing stroke volume. And this would also add in a little bit to our total peripheral resistance as well. Finally, epinephrine also acts on the heart. The heart also has adrenergic receptors that respond to epinephrine in similar ways to how they respond to our norepinephrine coming from the sympathetic branch of the autonomic nervous system. So epinephrine is also increasing your heart rate and stroke volume. So when epinephrine is present, we again see an increase in all three factors that contribute to our mean arterial pressure, right? So heart rate, stroke volume, and TPR are all going up here. Next, vasopressin and angiotensin 2 do affect our mean arterial pressure largely because they act as vasoconstrictors, right? So vasoconstriction, squeezing down arterioles, is increasing your total peripheral resistance and increasing your mean arterial pressure. They would also play a little bit of a role in regulating and responding to what's going on with the kidneys. So they do have additional roles to play here beyond this kind of simple vasoconstriction level, but we'll leave it there for our purposes since we haven't talked about kidneys yet. We also want to know that those arterial baroreceptors in the aortic arch and in the carotid sinuses are not our only types of baroreceptors. So we mentioned these briefly when we talked about veins. I think it was in that packet. But we also have low pressure baroreceptors. Okay. So the arterial ones aren't really under low pressure. Arteries are in general under high pressure. These low pressure baroreceptors in veins, right? So we've got some in our large veins, and we also have some in the heart, in the atrium. So what these are really doing are sensing more kind of blood volume. We often think of them as volume receptors, but they're going to work alongside those baroreceptors in the arteries. So when you sense low volume, that's also going to go kind of hand in hand with low pressure, right? So if they sense low volume, we're kind of in a hypotensive situation again. Yep. So when we see decreases in blood volume, this is going to be probably the same time that we're seeing decreases in MAP in the first place. So we're going to see a response that parallels and assists the baroreceptor reflex in pulling our mean arterial pressure back up, right? So this would ensure that in a case where that pressure drop is because, say, you're hemorrhaging, right, and you have lower volume in your body, right, this would tell your body something about that. And even if your problem is that you have less blood in your body, right, you still want to raise your blood pressure a bit because you want to make sure you have enough pressure to get blood up to the brain and to feed the heart muscle. There are a couple of other things that can affect our, I don't know, like our heart and our mean arterial pressure. So we have some links to breathing, some links to the concentrations of different molecules, particularly carbon dioxide, right? So what we're going to talk about here. So our chemoreceptors, as well as some responses to body temperature and exercise. So we'll take a quick peek at those. Okay. So respiratory sinus arrhythmia isn't something that necessarily happens to everyone. And according to your book, we don't totally understand why exactly this happens. But it happens to be the case that in some people and kind of more commonly in children, when you breathe in, so during inspiration, your sympathetic activity goes up a little bit. So the sympathetic branch, your autonomic nervous system, it's upregulated, which means your heart rate also goes up a bit. And the opposite happens kind of when you breathe out, right? So during expiration, during breathing out, your parasympathetic activity increases instead. So you see a drop in heart rate. So this is why it would count like as an arrhythmia. So an arrhythmia is like a change, an abnormal change to your heart rate, right? So it's not, yeah, your heart rate doesn't typically in a textbook vary if you're breathing, but we observe that in some people it does in real life as you breathe. Yeah. So some people use this and try to, when, they're trying to have target accuracy when they're shooting or something, try to alter their breathing to do it when your heart rate is a little lower, when you're kind of calmer and you don't have quite as much kind of potential for shakiness or blood changing how your hands are. Okay. The other kind of upside of this would be like if you feeling really anxious and you want to relax, you might try this out. Just try breathing out a lot. See if that helps you out. See if that helps you get more into a kind of parasympathetic state. Okay. So the chemoreceptor part is responding to components of the blood, particularly your blood gases, particularly carbon dioxide. Okay. So in response to these carbon dioxide levels, we know that we have our smooth muscle in arterioles is responsive to carbon dioxide. When we talk about lungs, we'll see that our bronchi are responsive to carbon dioxide. When we're talking here, we're talking about some receptors in your brain that respond to carbon dioxide, right? So we know that carbon dioxide is a waste product from metabolism. And we'll also see that carbon dioxide kind of acts as an acid in your blood. It can get converted into an acid. And so it would affect your blood pH, which is why your body really needs to make sure that we're keeping the carbon dioxide levels kind of low enough. Okay. So from the perspective of our mean arterial pressure, we're going to see that overall, we tend to see an increase in mean arterial pressure. Okay. Our bigger effects will come later on when we think about ventilation. Okay. So when we see these increases in carbon dioxide, we're going to see kind of two things, right? We're going to change our breathing, and we'll talk about what that does to exchange of the blood gases. But at the same time, we're going to be seeing an increase in total peripheral resistance as an effect of the chemoreceptors sending out signals through your body. We're going to see a decrease in heart rate. Now, this can be a little bit surprising because we've talked about how when we have metabolically active tissue, we want to send more blood there, right? So initially, this drop in heart rate doesn't totally make sense. But when we think about it from the perspective of making sure that those carbon dioxide levels in the blood stay low enough, we want to remember, or we want to learn, that your heart muscle itself produces carbon dioxide as it contracts, right? It needs energy, it uses energy in order to contract. So what this is actually doing is that carbon dioxide level in the blood is kind of taking precedence here over some of our other effects. And we're decreasing our heart rate a little to stop some of that extra carbon dioxide production. So overall, the effects might be a little variable here, but on the whole, the tendency is that this increase in total peripheral resistance in response to triggering those chemoreceptors more than makes up for the fact that we have a decrease in heart rate. So this is the one place that I can think of that we have two factors that are going in kind of opposite directions, right? By which I mean, if we're trying to think of which direction our map is going to go overall, right? We know we're paying attention to heart rate, stroke volume, and TPR. And for everything we've talked about up to this point, we've got a rising tide raising all boats, right? So if one thing's going up, all the others kind of are at the same time. So this is the one place that we want to remember. We might have a drop in heart rate at the same time as this rise in TPR. Overall, probably still an increase in mean arterial pressure. I'm just pointing this out because I think I told you before that pretty much all the time, everything goes together in the same direction. This is kind of our exception. Okay. Now, finally, we have our thermoregulatory response. Okay. So the setup here is that in addition to regulating MAP, regulating our blood pressure, your body also regulates its temperature, right? That's another homeostatically regulated variable. And your body temperature is more important to your physiological control systems than this mean arterial pressure is, right? So our thermoregulation, this temperature control, is going to take precedence. Okay. So this is being regulated by your hypothalamus partially. Okay. So when we notice that body temperature goes out, right? You're outside on a really hot day, doing really strenuous activity, you're overheating, we know, and we're going to start sweating, right? We can immediately see that kind of mechanism in place, trying to cool us down. Okay, so at the same time as we see that, right, in order to lose some of that heat that you're producing or soaking up from the sun and the environment, the way to increase heat loss is to make sure that that hot blood is coming to the surface, right? So you're overheating blood. We want it to flow to capillaries in the skin so that we can lose heat to the environment, especially assisted by that sweat, that evaporative heat loss kind of cooling you down. Okay. So to do this, to get that blood there, we have to vasodilate our blood vessels going to the skin, right? So we're increasing our flow to the skin, right? We're doing this by decreasing sympathetic activity, right? Because sympathetic activity does the constriction. So this vasodilation overall is dropping our total peripheral resistance as these blood vessels get bigger and wider. Flow is getting rerouted to the skin. Yep. So this is going to result in a decrease in your mean arterial pressure here, right? So if you see someone who's experiencing like heat stroke or something, you can see how this kind of works. Part of that is that we get a little lightheaded as our blood is flowing to the skin to keep us cool. We're kind of experiencing the negative trade-off here that our blood pressure changes at the same time. Okay. So most of our other factors, we've been really talking about this increases MAP, this increases MAP, this increases MAP. Here's an example of something that pulls your blood pressure down, decreases your mean arterial pressure. All right. Finally, we got a little note for ourselves on what happens overall when you're doing some light exercise. All right. So we're going to notice, all right, that our overall effects here is a slight rise in mean arterial pressure, possibly less than you'd expect, less than I expect when I'm going on a jog. Okay? So what's happening when you're doing some light exercise, right? So one thing, as you're exercising, we know our heart rate is going up, right? So our heart is beating faster, or increasing our cardiac output. In addition to our heart beating faster, our heart is beating a bit harder, or increasing that stroke volume. All right, so here are 2 pieces of cardiac output. Okay. We know that we are the cardiac output was on here as well. Guess I could have just drawn an arrow. Okay. So we know that we're changing the distribution of cardiac output at the same time. All right, so that cardiac output isn't going to all parts of your body equally. In particular, we're seeing that blood is rerouted to our skeletal muscle and to our heart muscle to kind of help us with this. We're going to see a slight rise in end diastolic volume, so slight rise in blood coming back to your heart as well. All right. But we may see a drop in total peripheral resistance, right? So this might be dependent on some other factors, but if you're doing light exercise, probably we're seeing a drop in resistance as we're rerouting that blood flow, right? I know when we talked about uh, epinephrine before, we said that usually we get up to a point where total peripheral resistance goes up, but depending on your intensity, maybe not right now, right? So I guess this is another case where we have our cardiac output and resistance doing slightly different things. But some of it is mean arterial pressure is still going up. Okay. All right. So that's what we have for our vasculature, right? So we've thought now about what blood vessels look like in general. We know some of the things about the layers of what we would see in their walls. We know our arteries are thicker. They've got some muscle in them, but they've got a lot of elasticity to them. They act as a pressure reservoir with their low compliance. Right? So we've talked about those a bit. We talked about our arterioles and how they're really key to regulating our resistance, our total peripheral resistance, which is a factor that, responds and can change potentially our flow, our cardiac output, but more often changes our mean arterial pressure. We talked about capillaries, where we have exchange happening, particularly exchange of fluid, which we can now kind of calculate and predict based on our net filtration pressures, our NFP. We walked through the veins. We know that they have to fight against gravity to pull blood back up to the heart because they're under a relatively low pressure overall. So we talked about some of the mechanisms for that. Talked about the fact that we also have a lymphatic system. It plays a role in returning some of the fluid that we lost through filtration, returning that into your bloodstream. Then we've linked it together, thinking about our effects on mean arterial pressure, which is basically a weighted average of our blood pressures, right? Weighting that diastolic pressure twice as much as our systolic pressure our systolic pressure. More diastolic, less systolic, because our heart is in diastole for a greater period of time. Okay. So now we're going to move on to what's in the bloodstream. Sorry, what's in those vessels. Okay. So we want to understand the different components of the blood. We're going to talk about its composition. And we're going to talk about some of the different types of blood cells we see in our bloodstream. So we're going to talk about our erythrocytes, our red blood cells, and our leukocytes, our white blood cells. And then we'll talk about clotting. So we'll talk about platelets and our hemostasis, so keeping our blood the same. Then we'll touch in a tiny bit thinking about disease. So here we see what it looks like if you take a vial of blood and then separate out its different parts, right? So we know that our blood contains cells. We're going to see it contains some platelets, which are like fragments of cells. It also contains a lot of fluid, right? So that fluid is our plasma, one of our two types of extracellular fluid. Okay. So on average, textbook person, textbook woman would have about 5 liters of blood. Man would have about 5.5 liters of blood. This can vary, right? But we would expect these percentages to say vaguely the same, right? So proportionally, you're going to expect there to be more plasma, right? And in this case, in our textbook, we have plasma as 55% of our blood volume. We're going to expect the next largest component to be our red blood cells, so these erythrocytes. It's about 45% of our blood volume. And then a tiny bit of that blood is going to be made up of super important things, just don't take up too much space. So our white blood cells are those leukocytes, and the platelets are these fragments of cells that are going to kickstart our clotting process when we experience an injury. If you're measuring these things in a lab or a clinic, one of the numbers that you're going to see is the hematocrit. So that's really telling you something about this erythrocyte kind of count here, right? So there are normal ranges for the hematocrit. You can see they both cross this 45% that we're picking as our number for our textbook to expect here. Okay. What you see here is kind of how we make blood cells. So we're going to see that blood cells get made in bone marrow, and they have to specialize into different types of blood cells. Ultimately, we'll spend most of our time talking about the erythrocytes, the red blood cells. You can see they're pretty different than a lot of these other cells that we see here. All right, so if we're looking at a stem cell that can produce blood cells, we'd call it a hematopoietic stem cell. So anytime you see like heme, you're gonna start to think about blood and blood cells. All right. So hematopoietic stem cell could become any of these different types of blood cells, but it's going to specialize and go down these different routes to get to different places. Right? So it could become a red blood cell over here, right? Could become one of these fragmentary platelets, right? And then we have a variety of different types of white blood cells that we have as options as well. Okay. Before we think about that, the extracellular fluid that all of these blood cells are going to be floating around in, the plasma that makes up about 55% of our blood, is going to be mostly water. So about 90% of the plasma, the extracellular fluid, is water. You're going to have 6 to 8% of proteins kind of swimming around in it. And we're going to have some electrolytes. So we're going to have pretty high concentrations of sodium and chloride ions. And we're going to have lower, but still very important protons. So this is going to contribute to the pH, right? The H in pH is this hydrogen ion here. We're going to have bicarbonate ions. We're going to have potassium. We're going to have calcium. And we've talked about the importance of the potassium and the calcium before, and we'll get back to these parts in our next unit. So let's first think about our erythrocytes, our red blood cells. Okay, so we got about 5 billion of them in every milliliter of your blood, right? So you've got 5 or 5.5 liters of blood, and then 5 billion red blood cells in every milliliter adds up to an awful lot. Okay. So their job overall is going to be to participate in gas exchange. So red blood cells are very critical in carrying oxygen around your body. They do also play a role in carrying away some carbon dioxide. And they're specialized in order to help this work better. One of those specializations is their shape. Right? So we refer to them formally as a biconcave disc. I think of them kind of like as a donut with the hole a little bit filled in. What this does is it gives them a lot of surface area, right? So they have surface area, surface area, surface area, right? Relative to their volume. Okay. So this helps stuff diffuse, right? So when we think about diffusion of gases, gases can cross a cell membrane, no problem. They're small enough, they're nonpolar, but they do still have to come in contact with a cell to do that. So when you've got a lot of surface area for your cell, this means we have a greater likelihood that a gas molecule is going to kind of bump into us and come inside. So this is influencing diffusion, both coming in and out of these cells. And they're pretty small, so about 8 microns in diameter, about 2 in thickness here. And that's going to let them go into small vessels like your capillaries. They're also a little squishy, so their membrane is particularly flexible, letting them travel throughout your body. But in order to create this shape, this size, to help them do their major functions better, we have to get rid of a lot of stuff that's in a normal cell. So if you look back at one of those pathway maps of how our hematopoietic stem cells become some specific type of blood cell, we're going to see that the erythrocytes got like a lot smaller at the end of the process. And this is because they lose their nucleus and their organelles along that differentiation pathway. Okay. So inside a red blood cell, you're not going to see all that much. You're not going to see mitochondria. They're not going to be doing some of the important metabolic processes that we've talked about before. Right? So they can't do oxidative phosphorylation really without a mitochondrion, right? And they're not going to be doing all that much glycolysis either. They're really going to be at a loss for a lot of ATP here. Right? So they can carry a lot of oxygen, they can carry a lot of carbon dioxide, but they're not going to be able to like fix themselves or do much at all in terms of cellular processes without these important components, right? So we're going to have to replace them a lot. So let's take a peek at how they're carrying their oxygen and carbon dioxide. So we're going to have to make these hemoglobin molecules earlier in the life cycle of a red blood cell, right? Because we lost the nucleus, so we've like lost our genes and stuff in our mature red blood cells. So we had to make this before they became a mature red blood cell. But we've got this cool protein called hemoglobin. Right? So hemoglobin has 4 pieces to it. So here we see our 4 subunits, right? So they got 2 in front in darker red and 2 in back in a lighter red. Okay, so these are the subunits. Okay? And each of those subunits consists of a globin part. So that's the part that's really the protein part, right? So that was a string of amino acids, a polypeptide that then folded itself in all these cool shapes, making this globular globin piece. And then we have the heme group nested inside. So here's our heme group. And this heme group is why you need iron in your diet in particular. So this heme group is centered around an atom of iron here. And we'll talk about iron and how we digest it and move it into our bloodstream and how we use it and recycle it later on. But we're noticing this. Okay. So you can move oxygen and carbon dioxide a little bit without hemoglobin, but it's hard and you can't put all that much into the plasma on its own, right? It gets saturated pretty fast, okay? So overall, when you think about oxygen in your bloodstream, almost 99% of it is bound to hemoglobin, binding into these heme groups, okay? Only about 1.5% is just straight in the plasma. So this is why you want to make sure you have enough red blood cells, why you want to make sure those red blood cells have enough hemoglobin in them. It's also this hemoglobin that's really giving blood its color. So when it has oxygen attached to the hemoglobin, it looks a brighter red. And when it's deoxygenated, doesn't have any oxygen bound in, it's a darker red. So not actually blue like the color of veins on a diagram, but that bright versus dark is some of what we're seeing when in the cadavers we see a difference between blood vessels. That darker red peeking through the vein walls is the thing that may look blue through your skin, potentially. Okay. It's also binding some other things. So it's not just oxygen that can bind into hemoglobin, although that's usually what we're concentrating on. It does also bind some carbon dioxide. It binds some protons, so some hydrogen ions. And it does bind carbon monoxide as well. The carbon monoxide, we do think about a little bit more often because ideally you don't have a bunch of carbon monoxide in your blood because hemoglobin prefers carbon monoxide to oxygen. So this is why you have monitors for carbon monoxide. It's because they will go and hog all the spots in the hemoglobin where oxygen would usually bind in, which means your body's not going to get oxygen. This is why you can die of carbon monoxide poisoning. Okay. So part of why we can carry carbon dioxide, which we would do some in our red blood cells, is because of this enzyme called carbonic anhydrase. Okay. So we're going to think about here about that acid aspect of carbon dioxide. So what we see here is this reaction between carbon dioxide and water catalyzed by carbonic anhydrase, which you would have in a red blood cell, and you would have just like in your body in general, gives us some carbonic acid, which then can dissociate into hydrogen ions and bicarbonate ions. Okay. Overall, the effect can be to make your blood particularly acidic. So that hydrogen ion piece plays a bigger role here than the bicarbonate ion does, largely because for our normal blood pH, we have an expected balance between hydrogen ions and bicarbonate ions, and this really throws it off. So this is critical for transporting carbon dioxide. We do transport it sometimes in these converted forms, but we're going to see later on that we're also really trying to keep it in balance here. So when we talk about our acid-base balance, which I think we'll be doing in our next unit as well, we're going to remember this aspect of carbon dioxide and how it reacts with things in your blood. Okay. So thinking a bit more about those erythrocytes, those red blood cells, right? They don't have a nucleus, they don't have DNA, they don't have organelles, they also don't have RNA, right? Because you don't have any DNA to pair with that RNA, really. So there's a lot of stuff we can't do. We're not doing a bunch of metabolism. We're also not dividing, right? We're not doing mitosis. We're not making copies of ourselves if we're a red blood cell, right? So we're going to have a pretty short lifespan in the scope of things. Any individual red blood cell is only going to last for about 120 days. On any given day, though, you're probably going to be replacing 2 or 3 million red blood cells per second because of how many red blood cells you have, right? So that's about 200 billion red blood cells per day that you're trying to take out of the bloodstream and get rid of, as well as making new ones. So we're going to be synthesizing our new red blood cells in our red bone marrow. So we're thinking like inside a long bone, you have bone marrow where we see these stem cells that are able to produce red blood cells. So including their earlier stages before they're fully mature red blood cells. So like along the way, we'd be producing hemoglobin. We'd have larger cells that still have a nucleus, DNA, RNA, organelles in order to do that before these red blood cells mature and then come out into your blood. Okay. So the process of making these new red blood cells or erythrocytes is called erythropoiesis. So making new erythrocytes here. Okay. So they're coming from the bone marrow. That's where they're born. And then they're going to go die in the spleen and in the liver. So we're going to be filtering them out in your abdomen. Your spleen is going to be about here. So on your left-hand side, kind of right below your rib cage, liver on the right, also see some recycling of our red blood cell components in those locations. Okay. So on the beginning side, the synthesis side, the creation of our erythrocytes, we're going to see this hormone erythropoietin is when it's going to be directly stimulating those erythrocytes synthesis, right? So this is the signal we're going to be getting from our kidneys telling us to produce some more erythrocytes. They're going to be developing also along their course through this other hormone, our hematopoietic growth factor is going to be helping them develop. This overall signal is coming from the kidneys when we start to see lower oxygen levels in the blood that's going through the kidneys. So there's a lot of like blood monitoring going on in your kidneys. This is one of the pieces that they're monitoring. So if you don't have enough oxygen in your blood, a way to solve that is to have more hemoglobin. Where do we put our hemoglobin? Red blood cells. So that's kind of the logic here. Okay. So we are differentiating those hematopoietic stem cells in our red bone marrow into erythrocytes when we get this signal from the kidneys through this erythropoietin. And those erythrocytes are going to make their hemoglobin, and then they're going to lose their nuclei and organelles and filter out into the blood in their small, kind of pared down form. If you were doing blood cell counts and trying to look for problems in a clinic, you might be looking for, sometimes we see cells escape into the blood before they've lost nuclei and organelles. So if you look in blood streams, you might find some of our like reticulocytes that still have those components. So this is something you could monitor, but this is what we expect to happen. This is what's normal and what we want to happen. Okay. So you're going to need some nutrients in order to make these erythrocytes, to make these red blood cells, right? So one of these, you need the iron for the heme group. Okay. The other pieces that you're going to need from your diet are vitamins and minerals that help you with cell proliferation, right? So we need to pack these red blood cells full of hemoglobin, and we also need to make more of them, right? We're making billions every day. So we'll stop here for right now, and we'll go through the life cycle of these blood cells on Wednesday. White blood cell stuff.